Hello students and welcome to the genetics series. Today we'll be looking at a second module, and the second module has to do with Mendel's Laws. So go ahead and subscribe and like the channel. This is Gregor Mendel, the man of the moment, the man who brought about Mendel's Laws of inheritance. So Gregor Mendel was born 20th July 1822 in the city of Exeter in Austria then, and he attended the University of Vienna as well as the University of Olmutz. So what's his major discovery? Mendel discovered the basic principles of heredity through his experiment in his garden, which has become the foundation of modern genetics and the study of heredity. Sadly, Mendel died January 6, 1884 in the city of Brno. This was in Austria then, but presently in Czech Republic. So before going further, let us define some terms. The gene. The gene is the unit of heredity or inheritance. This could also be looked at as a piece of DNA on a chromosome that contains all the coded information that controls a particular trait. Dominance versus recessive. Dominance is a situation whereby a trait or a character prevents or marks the appearance or the expression of another trait, even though the genes that are responsible for both of them are present. So if the factor or the gene expressed is dominant, is presented by a capital letter, and the gene that is prevented from expressing itself is said to be recessive and is usually presented by small letters. The genotype versus the phenotype. Now, the genetic composition of an individual is said to be his genotype, and it's usually presented by alphabets as shown here. Meanwhile, the outward expression of the genotype is the phenotypes. The phenotype could be color, in terms of height, short versus tall, a horned cattle versus one that is not horned, and also color coat of cattle. Alleles. Alleles are the alternative forms of a gene or a factor. It could also be looked at one of a number of possible alternative forms of the genetic information at the gene locus. A number of a set of genes that are on the same location on a given chromosome is also said to be allelic to that gene. So these are allelic to a gene represented by capital A also, but recessive form of small a. Homozygous versus heterozygous. Having identical alleles at the locus or at several loci is referred to as homozygous. So they're usually presented by the same letter, which are similar. The character that is homozygous is said to be homozygote for the trait or character. An individual rather is said to be homozygote for the trait or character. Now, this homozygous trait could be either in a dominant or it could be in a recessive form. It could also be double homozygote, the dominant state, double homozygote in the recessive state. Heterozygotes, on the other hand, have different alleles at a locus or at several loci. Hetero means different. So we have this heterozygote, and this is also heterozygote. Now, it could be single heterozygous, or it could be double or heterozygous. Single in the sense that only one trait, and double have two different traits. So Mendel carried out his experiment on a common garden pea, here represented. Okay. He selected the garden pea basically because he noticed that it was easy to cultivate, which is self-fertilizing, and it possesses several contrasting characters. Mendel studied 7 of these characters. So these are the 7 characters that Mendel studied. The garden pea studied the seeds and the whole plant. And on this column, you have the numbers that you observe in the F2 generation and the ratio that was obtained. We'll come to this later. So, considering the seed shape, we studied round seed versus wrinkled seeds. The color, we studied the yellow seeds versus green seeds. The flower color, we studied purple flower color versus white flower color. He also looked at the position of the flower, the axial versus the terminal. He looked at the plant height. He studied tall plants versus short plants. He looked at the pod shape, the inflated or full pod shape versus the constricted pod shape. He also looked at the pod color. So we have the green versus the yellow color. So he studied these characters separately by crossing 2 varieties with contrasting characters. And his experiment gave rise to 2 fundamental laws. These are referred to as the laws of genetics, or simply Mendel's laws of inheritance. Mendel's first law is known as the law of segregation, and it states that the 2 coexisting alleles of an individual, for each trait, we segregate, that is, they'll separate during gamete formation, so that each of the gametes gets only one of the 2 alleles. These alleles will unite again at random during fertilization of the gametes. We use this to illustrate Mendel's first law. So here we have the parental generation, okay, or segregating at the color, okay. So these are the gametes that are formed, okay. These are the gametes. These are the gametes that are formed. Now, going into reproduction, only one of the gametes will go into reproduction, but then they reunite again at random during fertilization. So if you look at this cross, or this example, we have a white plant that is being crossed with a red colored plant. And at F1 generation, all the plants are red in color. This is to signify that redness is dominant over whiteness. Now, when the plants produced in the F1 are self-pollinated, we have the gametes and the other gametes. In the F2 generation, Mendel obtained a ratio of 3 to 1. So the same thing is illustrated by tall versus short plants. So here tallness is dominant over short plants. This is the parental generation, and this is the F1 generation. Because tallness is dominant, all the plants are all tall, and when this plant is self-fertilized, the F2 generation will give us a ratio of 3 tall plants is to 1 short plant. To be noted that 1. All the F1 progenies in the first cross are red, and this is because redness is dominant. When the F1 yourself, that is self-pollinated, the resulting F2 progenies show a genotypic ratio of 1:2:1. That is to say that although all the plants appear to be red, there's only one pure red. 2 of them are heterozygotes and so are hybrids, and then there's only one pure white. But phenotypically, when you're looking at it, based on what you see, you have 3 that are red and you have 1 that is white. This is known as the monohybrid ratio, and this results from a monohybrid cross when only one gene locus is considered. And importantly, there is complete dominance. A test cross is usually carried out to find out the genotype of a plant or an animal. And this is usually achieved by crossing it, that is the plant or the animal, to a homozygous recessive individual, usually referred to as tester. Let's consider the test cross case below. We want to confirm if a plant that is phenotypically tall is homozygous tall, meaning the plant looks tall. We want to be very sure if it is homozygous tall, pure tall, or if it is heterozygous tall. So, if it is homozygous tall, it will carry the genotype capital T, and the tester, which is recessive, will be a short plant, small t. So the gametes that are produced from these 2 parents will be this and this. So if we take this into this square here, you see that when we cross these 2, we get this. We cross this, we get this. When we cross this, we get this. And when we cross this, we get this. Now, how do we confirm if the parent that is suspected to be homozygous is actually tall? If all the F1 progeny, the genotype is capital T, small t, that is, they are 100% phenotypically tall, then the suspected plant is actually homozygous tall. Two, if the plant suspected is heterozygous tall, then we expect a different ratio in the F1. So this is a heterozygous tall plant, which is crossed to a tester dwarf plant, and these are gametes. So we have this, we have this. So when you cross this, you get this, you get this, you get this, you get this. So if 50% of the F1 progeny is tall, phenotypically tall, and 50% is short, then the suspected plant is actually heterozygous tall. Now we can test to see if Mendel's ratio of 3:1 is actually true. Recall that Mendel counted the number of plants based on the character that he studied, and we saw the ratios earlier. But we can actually test if such a ratio is true by carrying out a test of Mendelian ratio. Now, this test tests the hypothesis using what is referred to as chi-square statistics. So let's consider a mating involving 2 heterozygous tall garden pea plants which produces two phenotypic classes. We will actually use the number observed by Mendel in his original experiment. So from that experiment, two heterozygous plants, when crossed, will give us this phenotypic ratio. We have tall versus short. And in his experiment, he observed 782 tall plants in the F2 generation and then 277 short plants. We want to be sure whether this ratio actually agrees with the expected Mendelian ratio. The expected Mendelian ratio is 3:1 for one single character. So what we want to know is if this observed number 787 to 277 contradicts or actually agrees with Mendel's monohybrid ratio of 3:1. So we are going to look at two hypotheses that we postulated and tested. We have the null hypothesis, which simply says that the observed number is equal to the expected number. That means the 787 and 277 tall and short lambs observed by Mendel is actually what is expected. And if that happens, this agrees with the ratio of 3:1. Then we have the alternate hypothesis, which says that the observed number is not the expected number. And if this happens, it means it is contradicting the ratio of 3:1. So looking at the plants counted, the total number of plants is 1064. This you do it by just adding the tall and the short plants. Now, with the null hypothesis, we will expect the following number of plants. So with the null hypothesis, we expect that the number of tall plants should have been 798, while the number of short plants should have been 266. Now, in chi-square, we test the discrepancy between what we observed versus what is the expected number of plants. And this is done using the formula chi-square is equal to the observed minus the expected squared divided by the expected. This is a short form of writing the same formula. So if we do that and we run the test by this, we will get a value of 0.607. Now the decision rule is to check the chi-square table under n-1 degree of freedom with 95% confidence level, and what we will get there is 3.841. 3.841 you observe is greater than 0.607. The implication is that there is no significant differences between the observed and the expected number. Why? Because this value is less than this value. The calculated value is less than the tabulated value. Therefore, we are going to be accepting the null hypothesis and we will reject the alternate hypothesis. That means that the observed number of tall and short plants actually agrees with the monohybrid ratio of 3:1. This is a confirmation that Mendel was actually right. The second law. The second law is known as the law of independent assortment, and it states that alleles of different genes we assort, that is, they will separate independently of one another during gamete formation. We're going to illustrate this by looking at the coat color and tail length loci in cats. Now, the coat color in cat is indicated by capital B, which means it is brown and it is dominant, or small b, which is white recessive, and the tail length is indicated by capital S, meaning short tail length is dominant, as against small s, long tail length, which is recessive. So a cross between a short tail length and white cat and a long tail length and brown cat will give us an F1 ratio of short tail length and brown cat. Now, these are the when this is self, when the F1 progenies are mated, you get these are the gametes that will be produced, and these are the F2 progenies. And in the F2, you will see that there's a ratio of 9, 1, 2, 3, 4, 5, 6, 7, 8, sorry, 1, 2, 3, 4, 5, 6, 7, 8, 9 cats that are brown and have short tail length. Then you have 1, 2, 3 that are white or short tail length, and you also have 1, 2, 3 that are brown but with long tail length, and they have only one that is white and long tail length. So the result at the F2 generation, you have 9 that are brown short tail length, you have 3 that are white short tail, you have 3 that are brown long tails, and you have 1 that is white and long tail. So the phenotypic ratio is 9:3:3:1. And this is a case of a dihybrid cross involving 2 contrasting characters. So we can also carry out a test cross where 2 different characters are involved. Again, we are trying to ascertain the genotype of an unknown plant or animal. For example, let's take a look at the expression of round yellow phenotype, and this may be represented by any of these genotypes. Any of these will give us round yellow phenotype. So if an F2 round yellow plant is crossed with a tester that is wrinkled and green, the analysis of the offspring will indicate the exact genotype of the round yellow plant. So let's look at the test cross result from this particular genotype. That will be, it is round yellow, it's tested against wrinkled green. These are the gametes. Okay, and when you do that, these are the offsprings that we produce. 50% will be round yellow and 50% will be round green. The result is because it's heterozygote. So go ahead and produce test cross results of this, this, and this. Again, we can test Mendelian ratio of 9:3:3:1 to be sure whether it is obeying Mendel's law. So we'll use our previous example of a mating involving the brown long-tail cat and white short-tail cat. So recall that the parents are brown long-tail and the white short-tails, and this the parental genotype. These are the gametes. So all the F1 generation will be brown and short-tail, and these are the progenies in the F2 generation. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9 brown short tail. We have 1, 2, 3 white short tail. We have 1, 2, 3 brown long tail and 1 white long tail. So that's the phenotypic ratio, 9:3:3:1. So these are the genotypes and the individual ratios of the genotypes. These are the phenotypic expression of the genotypes, and these are the actual numbers. We have 315 that are brown short-tailed, 108 that are white long-tailed, 101 brown long-tailed, and 32 that are white short-tailed. These are the actual numbers observed in the F2 generation. Recall that the expected Mendelian ratio for a dihybrid cross is 9:3:1. So again, what we want to do is to agree or disagree with the observed number, whether this agrees with this ratio of 9:3:1. Again, two hypotheses will be tested. The null observed is equal to the expected. The alternate, the observed, is not equal to the expected number. So when we add all these together, we have 556, the total number of cats, sorry, not plants, cats, and with the null hypothesis, we expect the following number of cats. So for the brown short-tail cats, it's 312.75. For the brown long-tail cats, it should have been 104.25. The white short-tail cat should have been 104.25. And for the white long-tail cats, it should have been 34.75. So all you need to do is to take the ratio over the total number of boxes as 16 multiplied by the total, and you get this value. So again, chi-square test, we test the discrepancy between the observed and the expected using this formula. And if we carry it out, this is for the first one, the second one, the third one, the fourth one. And these are the values we get. And this is the final answer, 0.47. So our decision rule again is to check the chi-square table at 95% confidence level at n-1, that is 3 degrees of freedom. This is the value we get. We observe that this value is way greater than the calculated value. So we'll now agree that no significant differences exist between the observed and the expected number of cats, since the calculated value is less than the tabulated value. So the null hypothesis is again accepted and the alternate hypothesis is rejected. In conclusion, we say that because the observed ratio of cats based on color and tail length agrees with the Dihybrid ratio of 9:3:1. Again, Mendel was right. So, are the monohybrid and the hybrid ratios always obeyed? The answer to this is an emphatic no. And this is subject to our next module. So in our next module, we'll be looking at exceptions or departures from the classical Mendelian ratios. What are those situations that will lead to Mendel's laws of 3:1 and 9:3:3:1 not being obeyed? That is the subject of our next module.